but in cooler temperatures, the growing slows down or it stops altogether. That's not the case with Listeria. Even at these cooler temperatures, the organism will still grow, okay? For some reason, I'm waiting for my screen to advance. Hold on one second. Okay, finally. Okay, now here's another bit of information we need to know about the organism. It is a class of bacteria that is said to be gram-positive. Now, all bacteria fall in one or two classes. They are either gram-positive or gram-negative. Let me just explain to you what that means. When we say that a bacterium is gram-positive, if you look at the composition of the cell wall for that bacterium, it is very thick, okay? Gram-positive bacteria, once again, have thick cell walls, all right? Given that information, what do you think gram-negative bacteria are? Anybody, who can tell me? Given the information we know about gram-positive bacteria. Go ahead.
They have thin walls?
Yes, ma'am. That's correct. The cell wall for gram-negative bacteria are a lot thinner, okay? The composition for gram-negative bacteria is a lot thinner than what we see when we compare them to gram-positive, all right? Now, I alluded to the fact earlier that Listeria can grow at refrigeration. So anytime that we have organisms that can actually grow at cooler or refrigerated temperatures, we call these psychotrophic organisms. Excuse me. Literally, what this term means right here, when we say that something is psychotrophic, that means that these organisms can tolerate the cold. I'll repeat that for you. If we say that an organism is psychotrophic, that means that they are cold tolerant. They can tolerate cold temperatures. So in the case of Listeria, we see here that can grow from temperatures that are from anywhere from 0 to 45 degrees Celsius, okay? Now, if you were to, if you were to introduce this bacterium to temperatures that were slightly higher, then guess what? The growth rate is gonna increase significantly. But this can be said for all bacteria. Okay, so once you introduce bacteria to cooler temperatures, the growth will either slow down or it will stop altogether. But the cold will not kill bacteria. Let's be clear of this. Colder temperatures will not kill bacteria. It will just simply slow down their growth or stop the growth entirely. Now, if we wanted to kill bacteria, that's something completely different. To actually kill bacteria, you would have to increase the temperature significantly. So to adequately kill bacteria, we're talking about having to increase the temperature well above 55 degrees Celsius or even higher. So the more extreme temperatures that you expose bacteria to in terms of heat, the heat will actually kill these bacteria. But then you have some bacteria that can actually survive extremely warm temperatures, but we're not going to get into that. But just know that those numbers are few, okay? Now, still on the discussion of Listeria and other bacteria, we have bacteria that are said to be aerobic, and then we have bacteria that are anaerobic. When we have bacteria that are aerobic, that means that they require oxygen to grow, okay? Aerobic bacteria, once again, require the presence of oxygen so they can actually survive. Now, given that information, what do you think anaerobic bacteria are? Anyone want to take a guess? If we're talking about anaerobic, aerobic bacteria require oxygen. What about anaerobic?
They don't need oxygen.
That's
to
correct.
survive.
That is correct. Anaerobic bacteria do not require oxygen, okay? And in some cases, if you were to introduce these bacteria to oxygen, the oxygen more than likely will kill those bacteria, okay? Now, with Listeria, this particular organism is a facultative anaerobe. What this literally means right here is that it's an organism that is anaerobic. It does not need oxygen to thrive. But if oxygen was present, then guess what? It will grow a lot quicker, okay? The growth rate will actually increase if oxygen is actually present. And that's what a facultative anaerobe is. They don't need oxygen, but they do a lot better when oxygen is actually present, okay? Last detail that we should know about the bacterium itself is that in terms of the shape, it comes in a form of a rod shape. Now, I have a picture off to the right-hand side of the screen that gives an illustration of at least two Listeria monocytogeny cells. And of course, we see that it is actually rod-shaped, all right? It is a rod-shaped organism. But be aware of this fact. Bacteria typically come in three shapes. They can either come in these shapes right here, rods, spheres, meaning they are circular shaped, or spirals. Those are the three shapes that bacteria generally come in. So depending on what bacteria you're talking about or you're looking at, they would come in one of those three shapes. Either they would be rods, spheres, or spirals. Now, in terms of why Listeria actually causes disease, the specific disease that it causes in human beings is known as listeriosis. Now, we already mentioned this a few minutes ago, that the mortality rate or the death rate associated with infections is around 20%. But it's to say that, it can be as high as 30%. All right, so the range for Listeria deaths related to Listeria infections is somewhere between 20 and 30%. Okay. Now, when people are actually dying, this actually gets noticed. This is why people actually take it seriously. Now, when it comes to deaths that result from listeriosis, those who are most vulnerable are pregnant women, as well as the people who are immunocompromised. Now, in pregnant women are advised not to eat ready-to-eat meats or dairy products such as cheese because of the threat that Listeria might be in that particular product. The issue is that if they consume those ready-to-eat products and it's contaminated with Listeria, then it can actually cause them to miscarry, okay? It can actually cause those pregnant women to miscarry or cause stillbirths, okay? Now, the other group that is at risk would actually be those people who are immunocompromised. So you have very young children because their immune systems have not fully developed. That will be one group of immunocompromised people. Then we have the elderly, okay? Their immune systems aren't what they used to be especially when they were younger. And then we have those adults who may have some type of illness that compromises their immune system, such as those people who have HIV/AIDS, who have not sought treatment for that particular condition, okay? So those are the groups that are at most risk as a result of listeriosis, okay? Another thing we need to be aware of about listeriosis and Listeria monocytogenes is that it actually attacks the digestive tract, okay? Listeria, it attacks the digestive tract. That being said, what do you think the primary symptom will be associated with an infection by listeria? What do you think the primary symptom would be if it attacks the digestive tract?
Diarrhea.
Diarrhea, okay? That would actually be the primary, yes, the primary symptom associated with it. But not only that, but you have flu-like symptoms such as fever, as well as body aches that go along with listeriosis, okay? Now, with any type of bacterial infection, the most common treatment for bacterial infections, especially ones caused by listeria, would be antibiotics, okay? Now, and as a side note, you would not use antibiotics to treat viral infections because, once again, antibiotics are only essential against bacterial infections, but not viral infections. And the primary means of actually diagnosing a person who has listeriosis would be taking a stool sample from that individual, okay? They would actually test the stool sample, and if it comes back positive for Listeria monocytogenes, then the healthcare provider will provide some type of antibiotic treatment. The most common type would actually be ampicillin or penicillin. Now, mind you, these are not the only types of antibiotics used to treat listeriosis, but they're the most common, okay? So that is the background information that's related to Listeria monocytogenes. Now, given that information that we've just gone over, I'm going to go ahead and see how much you all were paying attention. So, Ms. Cole, all right. Now, given the information we know about Listeria, we know that it's gram-positive, right? What is a gram-positive bacterium?
It has a thick cell wall.
That is correct. And if we are talking about bacteria that are gram-negative, what does that tell us about the cell wall?
It has a thin cell wall.
A thin cell wall. All right. Let's see. Ms. Hampton, all right. What do we mean when we say that an organism is psychotrophic? What do we mean when we say that an organism is psychotrophic?
That it can stand like cold.
Yes, it can stand cold or cooler temperatures. They can tolerate the cold.
Mm-hmm.
Now, to piggyback on that, do cold temperatures kill bacteria?
No.
No. No, okay, you already answered that.
I'm sorry.
You're good. But no, cold temperatures do not kill bacteria. They just either slow down the growth rate or stop it altogether. Let's see. Mr. Fain, okay. What can you tell me in terms of the shape that bacteria come in? What are the three shapes that bacteria can possibly come in?
A rod shape, a circular shape, and then
what am I doing with my hand? I'm forming what type of shape?
a corkscrew shape.
Corkscrew or spiral shape? Yes.
Yeah, spiral shape.
All right. Okay. Let's see. What else can I ask you? Ms. Muhammad, what is a pathogen? What is a pathogen?
A pathogen is a type of bacteria.
Well, Okay, all right. It can be a bacterium, a virus, or okay, but what does a pathogen do?
Something that affects humans,
More specifically, what does it
causes
cause?
illness.
It causes illness or disease. All right. They are any agents that have the ability to cause disease. Okay. All right. Now, as I said before, I just put this PowerPoint together just about a few hours ago, but I will upload this after we get off of the talk. Okay. Are there any questions about the information that we just discussed as it relates to Listeria?
What are the cures for Mysteria?
Go, say that again.
What are the cures for Mysteria?
That first part sounded a little muffled. What? Say it one more time.
What are the cures for Mysteria?
Well, in terms of a cure, as I said before, it's a bacteria. It would be a bacterial infection, and with basically all bacterial infections, you would need to use antibiotics. Okay, so and this question came up in the last period. What if you're allergic to, say, penicillin? Okay, if you're allergic to a certain type of antibiotics such as penicillin, then what the healthcare provider will actually have to do is they'll have to prescribe another class of antibiotics. As I mentioned to you before, those two that were just mentioned, ampicillin and penicillin, those are not the only antibiotics that can be used to treat listeriosis, okay? But they're just the most common type, plain and simple. All right.
Thank you.
Okay, any other questions? Oh, go ahead, Ms. Jeter.
Okay, hi. To be clear about how the the illness is caused, it's by consuming foods that has like listeriosis on it.
Well, okay, yes, that's correct. But now we need to get straight on our terminology. Now, listeriosis is the actual disease. Okay, listeriosis is the actual disease, and the actual agent or the actual organism that causes the disease is Listeria monocytogenes. So if you consume a food product that it has a high count of Listeria monocytogenes in it, then the likelihood of you becoming ill is significant. All right. So again, but when it comes to the terminology, know that listeriosis is the actual disease caused by Listeria monocytogenes.
Thank you.
Yes, ma'am. Go ahead, Ms. Muhammad.
This is not exactly related to listeria, but why is it that penicillin and ampicillin are the most commonly used?
Well, generally speaking, when it comes to those two specific types of antibiotics, it just so happens that it's just a wide range of bacteria that they can actually kill. Okay, that's basically what it comes down to, is because they can kill not just one specific type of bacteria, but they can kill a wide range of bacteria. That's basically what it comes down to.
Okay, thank you.
Any other questions about what we just talked about as it relates to listeria and foodborne illness? Okay, so again, I'm going to post this for you all after we finish our talk. Now, what I also want to discuss with you is the fact that while we are in this course, what you'll also learn how to do is, and hopefully when we get back into the lab, you'll actually learn how to culture this bacterium. And when I'm talking about culturing the bacterium, I'm talking about actually growing it up in the laboratory, okay? But a part of actually being able to culture this bacterium, as well as other types of bacteria, we have to know how to use a certain type of technique known as aseptic technique. Now, literally, when we're talking about doing aseptic technique, we are specifically talking about trying to contaminate our sample as little or not as little as possible or not at all. We want our sample, for the most part, to be as pure as it can be without outside contamination. Okay, so what I want to do now is I actually want us to look at a video, and I actually posted that video earlier today that explains aseptic technique. So let me go ahead and pull that up for you. Can you all see this screen?
Yes.
All right, let's see if you all.
In this video, you will learn the proper practices and procedures.
So were you all able to hear the audio? Just to be clear, were you all able to hear that audio?
Yes.
Okay, let me just enlarge the screen.
Barely.
Yeah, I have it as high as I can go. But again, this is also, but it's posted on Blackboard though, under the content tab. But let's go ahead and look at the video, look at it, go through it.
For performing aseptic technique. Using aseptic technique, you will be able to transfer a bacterial monoculture into a fresh medium without introducing any contaminating microorganisms. Two of the most commonly used tools for transferring bacteria are the inoculation loop and the inoculation needle. Knowing how to use a Bunsen burner is an important skill in aseptic technique. To light a Bunsen burner, start by...
Okay, so why do you think you need the Bunsen burner?
Sterilization.
Okay, how is it sterilizing it? Because what did we just say? What does heat do? What does heat do?
High heat can kill pathogens.
All right, it can kill pathogens as well as other organisms. Okay, so remember, the higher the heat, the more heat that you actually use, then that means whatever organisms are present will actually killed off. Okay.
Closing the valve at the base by rotating the collar so that the vents are covered. Then slowly turn on the gas. Once you hear the gas, ignite the burner and set the height of the flame by adjusting the amount of gas. The temperature of the flame can be adjusted by rotating the collar to control the flow of oxygen. The coolest flame is yellow and orange. A medium flame is blue. And the hottest flame is a roaring blue flame characterized by a clear blue cone in the middle. The tip of the cone is the hottest part of the flame. Inoculation tools are sterilized by passing them through the hottest part of the flame. Once sterilized, inoculation tools should not be set down on any surface. In this section, you will learn how to transfer bacteria from a liquid culture to a petri dish. After flaming a loop, take the lid off the container of liquid culture and pass the mouth through the flame. Then dip the loop into the liquid culture and flame the mouth again before replacing the lid. Transfer the bacteria to your petri plate. Lift the lid of the plate and hold it over the base as a shield to prevent contaminants from falling onto the plate. Spread the bacteria over the entire surface of the plate in all directions. Rotating the plate a quarter turn between spreads will ensure that the bacteria is spread evenly across the plate. In this section, you will learn how to transfer bacteria from a liquid culture to a liquid. After sterilizing an inoculation loop, take the lid off the container of liquid culture and pass the mouth through the flame. Then dip the loop into the liquid culture and flame the mouth again before replacing the lid. Now take the tube of liquid media and pass the mouth through the flame. Then dip the loop with bacteria into the tube. Flame the mouth of the tube before replacing the lid. Flame the loop to sterilize. In this section, you will learn how to transfer bacteria from a Petri plate to a stab culture. Start by sterilizing an inoculation needle by passing it through a flame. Allow the needle to cool. You can test for coolness by tapping the needle in a clear area on your agar plate. While holding the lid as a shield, gently rub the inoculation needle over a single colony. Transfer the bacteria to a culture tube filled with nutrient agar. Remove the top of the tube and insert the needle, making 3 deep stabs. When finished, replace the lid on the tube and sterilize the needle. In this section, you will learn how to transfer bacteria from a Petri plate to a liquid. Sterilize an inoculation loop by passing it through a flame. Let the loop cool. You can test for coolness by tapping the loop in a clear area on your agar plate. While holding the lid as a shield, gently rub the inoculation loop over a single colony. Now take the tube of liquid media, remove the lid, and pass the mouth through the flame. Then dip the loop with bacteria into the tube. Pass the mouth of the container through the flame before replacing the lid. Sterilize the inoculation loop to complete the process.
Okay, so tell me what, by looking at that video, what stood out to you? What stood out to you in that video the most?
How much he like sterilized in between doing different things.
All right. So again, you want to try to make sure that you don't contaminate your samples at all. Okay, you want to have as pure a sample as possible. Okay, so notice how the gentleman was, every time that he opened up a container, he ran the top of that container across the flame, and then after opening it up, and then when he was done, he ran it back over the flame and closed it, as well as with the loop and the stab, flaming it before use and then flaming it after use. Okay, so that would actually be the recurring thing that we see with aseptic technique. So again, the whole point of aseptic technique is to make sure that we have as pure a culture or sample as possible. All right. So so that is, but hopefully later on in the semester, you'll actually be doing that, especially when it comes to culturing your bacteria. All right. Again, depending on whether or not the connection issues are rectified in Henderson, up until, so basically until further notice, we'll actually be doing the lab virtually. Okay, I don't want to do it that way, but it seems like we're going to have no choice. All right. Now, before we also end, just a couple of other things. Now, if you have not completed that first LAPSA simulation dealing with lab safety, you have until midnight until that closes. All right. So if you have not completed that particular simulation, you have until midnight. Also, there is a new LAPSA simulation that is also available, that is now available to you. It actually became available about 40 minutes ago. Okay, that particular LAPSA simulation is dealing with aseptic technique. That particular LAPSA simulation is due next Thursday. All right. So once again, the simulation, if you haven't done the simulation on lab safety, that is due tonight before midnight. After midnight, it will disappear. The one on aseptic technique is due next Thursday. Okay. Any questions? Nope.
Question.
Go ahead.
Okay, I apologize. I'm not sure if you said this already, but when is our in-person meeting?
See, that's the thing. Yeah, now we were supposed to have it today, but because the Wi-Fi connection in Henderson is so poor, there was no way that I could actually talk to people online as well as you all in person effectively. So that's the reason why I'm doing it this way. Okay, so, but again, until further notice, we'll meet virtually.
Thank you.
Yes, ma'am. Any other questions? All right. So if there are no other questions, make sure that you, again, if you haven't done the lab safety simulation, do that before midnight. And the aseptic simulation is also available to you, and it will be available to you until next Thursday. If there's nothing further, we will see you next week. So have a good day.
Have a good day.
Have a good day.
All right. Yeah, come on.